Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and Akoya Biosciences webinar titled Unraveling Immunotherapy Resistance in Advanced Melanoma, Novel Insights from Longitudinal Spatial Multiomics Studies. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Camelia Quek, who's the CINSW Research Fellow and Honorary Senior Research Fellow at the Melanoma Institute Australia and University of Sydney, who will present research on immunotherapy resistance in melanoma using advanced spatial multiomics and single-cell analysis. And with that, I'm very pleased to hand things over to Dr. Camelia Quek. Camelia, thanks so much for joining us today, and the floor is yours whenever you're ready.
Thank you very much for the warm introduction, Ilham, and also many thanks for the opportunity to share our work in today's webinar. For today's presentation, I'll be focusing on longitudinal spatial multiomics in melanoma patients treated with immunotherapy. To start, here's a brief background of our research. Advanced melanoma is where the tumor has spread beyond the skin to nearby tissues, lymph nodes, or distant organs in the body. In Australia, melanoma is often referred to as Australia's national cancer, as we have one of the highest rates of melanoma in the world. At present, there are no current therapies that can treat advanced cancer effectively. The common standard of care therapies to treat patients with BRAF-mutated advanced melanoma is targeted therapies, including BRAF and MEK inhibitors. In the last decade, anti-cancer treatments such as immunotherapies that include anti-CTLA-4 and anti-PD-1 are able to empower the body's immune system and restore the cancer-fighting ability of T cells to shrink tumors. How do the drugs work? Firstly, PD-1 monoclonal antibodies such as pembrolizumab and nivolumab block the PD-1 signaling pathway and thereby restore cancer-fighting ability of our immune system to eliminate the growth of cancer cells. Another treatment strategy is to implement combination drugs by blocking two inhibitory signaling pathways. In addition to blocking PD-1 signaling pathway, CTLA-4 is also to further restore the balance of the immune system so that immune cells, such as T cells, will be fully activated to detect and eliminate the cancer. Recent advances in treatments with BRAF/MEK inhibitors in COMBI-DV trial and anti-PD-1-based immunotherapies in CheckMate 67, Keynote 6, and CA184 trials have demonstrated survival benefits where the overall survival rates are higher than 50% when compared to EP alone. However, there is still a subset of advanced melanoma patients who either failed to respond or experience relapse after treatment. We hypothesize that there are a few reasons why therapy-resistant cells exist in the tumors. These reasons may be due to the genetic traits of the tumor itself or the extrinsic factor involving immune and stromal cells in the tumor microenvironment, or even the influx of immunosuppressive cells or cytokines that can hinder the T cell functions. In our recent work, we found that distinct cancer clones and cell types within the tumor microenvironment can impact patient-specific responses and immunotherapy outcomes. For example, in the first paper published in Cell Reports, we found that firstly, specific melanoma clones emerge in the tumors after combination therapy. Secondly, B-cell signature from lymphoid aggregates can differentiate immunotherapy response outcomes. Lastly, the loss of memory state in T cells can instigate the development of acquired resistance. In another example, the second paper published in Nature Communications, we found two distinct subtypes of immunotherapy-resistant young melanoma patients, one with higher infiltrations of T cells in the peritumor stromal region and the other with low T cell infiltration and non-immunogenic tumor microenvironment. By comparing the transcriptomic profiles of young melanoma patients, we are able to define a gene score that predicts the likelihood of resistance. Recently, there has been an interest in looking at tertiary lymphoid structures due to their association with good response outcomes in cancer immunotherapy. A number of papers also demonstrated that CD20 positive B cells are commonly found in association with tertiary lymphoid structure that actively help to modulate anti-tumor immune activity. Hence, it will be interesting to explore whether these lymphoid aggregates exist in responding and resistant immunotherapy patients. For the first part of the talk, I'll be presenting our current work on single-cell spatial multiomics in melanoma patients treated with immunotherapy in the metastatic settings. As I have mentioned earlier, there is still a subset of advanced melanoma patients who are not responding to immunotherapy. So in order to tackle immunotherapy resistance and tumor immune heterogeneity, we use a combination of single-cell technologies and computational methods. Here we have a subset of patients that have paired tumor dissociate and FFPE tissue. Each of the respective samples are passed to single-cell site sequencing that simultaneously sequence transcriptome and cellular epitome at the single-cell level. In addition, for fixed tissue, we use multiparametric phenolcycler imaging system, which is formally known as CODEX, that can stain up to tens or hundreds protein targets. As you may have already knew that the single-cell technologies have both strengths and weaknesses. For example, single-cell sequencing often provides full spectrum of the biological programs, but often requires tissue dissociate that results in the loss of spatial context. Multiplex imaging can provide cell location, but often will limited by the set of pre-selected markers that only capture the key cell types. As such, we rationalize that this could be harmonized by integrating these different modalities. In the past, there were no tools available for such data integration, so we built the integration toolkit by putting existing tools together to align data from single-cell transcriptomics, proteomics, and spatial features, along with the clinical features. I will briefly walk you through the data integration steps. After processing the raw data, a data matrix of each modality will be generated. We normalize each cell by total counts over all features and perform clustering. We then annotate each cell based on references, gene signatures enrichments, and clinically verified markers, especially for malignant cells. We next perform anchoring based on nearest neighborhood and use transfer learning method to map back the cells to the spatial location and relate those cells back to the clinical pathological features. The pipeline is now accessible in GitHub so that others can fork this pipeline and undertake similar research studies. In order to demonstrate the integrated results of the integration toolkit, we generated 3 different maps. The first map shows the cell populations at the molecular single-cell sequencing level, and the second map shows the key cell types in phenolcycler imaging. And lastly, the spatial maps show the different cell types in the tissue after integration. We demonstrated that this multimodal integration toolkit accurately aligned with the integrated data from different modalities by confirming tumor microenvironment markers across the data sets. An example showing here is the CD8 T cells showing CD3 and CD4 and CD8 across phenolcycler protein, site-seq at the protein level, and as well as at the RNA level. Our early findings from 5 patient tumors with advanced melanoma highlighted the key immune subset and tumor and their microenvironment cell populations. At the patient level, non-cancer cells, such as the immune cells, are represented by many patients. However, the cancer cells are very patient-specific. For example, there are patient-specific gene expression profiles that are very evident across patient MIA-10 and MIA-11. To put into perspective at how we have used our methodology in clinical samples, here is a case study of a patient with innate resistance. Patient MIA-11 is a 66-year-old female, BRAF wild type, with stage 4 disease and NRAS Q61L mutation, who failed to respond to mono or combination immunotherapy drugs, but has response to a new combined anti-PD-1 and lenvatinib drug, which target not only the immune checkpoint receptor, but also the multiple kinases and growth factors. At the cancer cell level, in these UMAP plots, we found that CDK4 and KRAS positive melanoma cells were persistently present at all time points. Additionally, MIF positive, SPARK-L1 positive, SEF, and STAT1 positive melanoma cells emerged after immunotherapy. Throughout the course of the treatment, as illustrated in the heat map, we observe a dynamic gradient of low to high expression of antigen presentation genes and inflammatory cytokines, and high to low immunosuppressive cytokines. The cyclic UMAP plot shows distinct cell types from the tumor microenvironment, and these cells are mapped to their spatial level. When we look at the tumor microenvironment, this patient has the correct molecular profiles with highly activated T cells that has the potential to respond to immunotherapy. However, at the spatial level, these highly activated T cells in blue and yellow are enriched in the peritumoral space, which is outside the melanoma cells, highlighted in red before treatment, as you can see in the immunofluorescent images. At time point 2, these T cells are concentrated along the tumor margin after anti-PD-1 based therapy. When the tumor responds to the new combination drugs at time point 3, an immune-inflamed environment is observed where lymphocytes are infiltrated into the tumor bed area. From the collective data, we understand that the tumor microenvironment is dynamic and can change over the course of treatment, and certainly there are potential in targeting the tumor microenvironment for effective cancer therapies. Furthermore, when we look at how the melanocytes behave with their surrounding cells, they were interacting with immune cells, fibroblasts, and blood vessels throughout the course of the treatment, highlighted by the red areas in the heat map. Except for CDK4 and KRAS positive melanoma cells that remained hiding in the tumor microenvironment by having minimum interactions with their surrounding cells, and perhaps a way to escape from immunotherapeutic drugs. In addition, the fibroblasts also express certain levels of cytokines that favors the growth of the stromal barrier, preventing the infiltrations of immune cells. Next, we are curious to see if there are any functional diversity in the tumor regions of T cells. We then compare the top genes between intratumoral and peritumoral T cells and found that the T cells confined to the stromal area had a lower expressions of chemokines than the intratumoral T cells. Although intratumoral T cells are present in the tumor area, but they have downregulated IL-7R that can impair IL-7 signaling, which can reduce proliferative capacity and cytotoxic function. As such, these results demonstrated a proof of concept that functional diversity exists between T cells in different regions of the tissue. In recent years, as I have mentioned in the previous slides, that there has been an interest in looking at tertiary lymphoid structures due to their association with good outcomes in cancer immunotherapy. Hence, we thought it would be interesting to explore whether these lymphoid structures exist in responding and resistant immunotherapy patients. We adapted the same data integration workflow as before. Here we show an image of a tertiary lymphoid structures or lymphoid aggregates in patients who are responding to immunotherapy, where we have seen the enrichments of CD20 B cells and CD3, CD4, and CD8 T cells at this area. We initially thought that lymphoid aggregates are only identified in responding patients. However, we also noted lymphoid aggregates in innate resistant patients, as you can see the enrichments of CD20B and as well as T cells in this region. Similarly, we also see lymphoid aggregates in tumor region in acquired resistant patient. Since the spatial level of lymphoid aggregates are similar in responding and resistant patient, we next look at the molecular signatures that comprise in tertiary lymphoid structures. We found that these molecular signatures are related to B cell immune profiles and express high in responding patients when compared to the resistant patients. When we validated these lymphoid structure signatures in the anti-PD-1 based therapy melanoma cohort and TCGA dataset, we found that the patients who highly express these lymphoid structure signatures have improved overall survival and progression-free survival outcomes. Overall, we think that the molecular compositions of tertiary lymphoid structures are important in understanding immunotherapy response outcomes. To summarize our findings, in the first part of this talk, this spatial multimodal integration study allowed us to firstly identify final cell subpopulation in the spatial tissue architecture using thousands of cell markers from sequencing to identify subpopulation of immune cells and map this subpopulation back to their spatial environment in the tissue. Secondly, this study allowed us to identify functional diversity between peritumoral and intratumoral T cells, which can have important implication in drug design by considering whether to target immune cells in the peritumoral stromal area or enhancing intratumoral immune responses to improve treatment outcomes. Thirdly, the study highlighted that the molecular composition of lymphoid aggregates can influence patient outcomes by identifying lymphoid aggregates associated B cell signature in patients who have higher survival rates as compared to those who have minimum or no expression. The next part of the presentation will focus on spatial immune microenvironment of stage 3 melanoma patients treated with neoadjuvant immunotherapy. Neoadjuvant immunotherapy is a treatment where a patient receives immunotherapeutic agents before surgery. This approach aims to enhance the body's immune systems against the tumor, reduce tumor size, and improve surgical outcomes. One of such trials is the PRADO trial. Patients with macroscopic stage 3B or 3C melanoma are enrolled and receive 2 cycles of 1 mg EP and 3 mg nivo over a period of 6 weeks, followed by index lymph node resection. There are 3 groups of response outcome. The first group refers to patients who have pathologic complete response or near complete response, have less than 10% viable tumor cells left. The second group refers to patients who have partial response, and they are often individuals whose tumors have shown 10 to 50% viable cells. And lastly, for those non-responders, there are more than 50% viable tumor cells left in the index lymph node. In this study, we collected a total of 26 fixed tissue blocks from PRADO trial. These samples comprise of a total of 17 baseline samples and 9 of the week 6 post-therapy samples. The phenolcyclofusion experiment is performed by my PhD student Katrin Bai, who has done a fantastic work in staining the tissue samples. We then examine their immune microenvironment by looking at a panel of 40 markers that comprise of antigen presentation features, lymphoid cells, myeloid cells, melanoma, immune checkpoints, blood vessels, stroma, and immune function. This is a representative image of a patient with pathologic complete response at baseline, showing CD14 macrophages, CD8 T cells, CD4 T cells, CD20 B cells, and CD31 blood vessels. At baseline, we observed that the complete responder had an overall high immune cells and patches of CD20 B cells aggregates in the tissue. Using the patient samples from week 0, we observed that this patient at week 6 after treatment not only has a high influx of immune cells, but also a structural resemblance of tertiary lymphoid structure enriched with immune activity and immune aggregate that strongly highlighted that the treatment is working for this particular patient. Next, this is a representative image of a non-responder at baseline, showing CD8, CD4 T cells, CD20 B cells, CD14 macrophages, and CD31 blood vessels. Melanoma cells are in purple color. In baseline, for non-responding tissue, we observe little or minimum CD4 and CD8 T cells and no B cell aggregates. It is still a work in progress at the moment to tease out whether these T cells are dysfunctional or whether tumors secrete suppressive cytokines that impede immune cell activity. Looking at the same patient at week 6 post-treatment time point, the tumor microenvironment still remains similar to baseline, where these T cells were dispersed and scattered around the tissue, but not infiltrating into the tumor area. Since our recent work and others have shown that the tertiary lymphoid structures are associated with good outcomes in patients treated with standard anti-PD-1 based immunotherapy, we next explore whether tertiary lymphoid structures are seen in the neoadjuvant patient cohort. This is an image showing the different type of structures that can be found in the tissue. The structures comprise of lymphoid aggregates, immature, and as well as mature tertiary lymphoid structures. So at baseline, we observed that the complete responder display an immature tertiary lymphoid structures showing enrichments of B cells in close proximity with CD8 and CD4 T cells. At week 6, the tissue from the complete responder further demonstrated a structural resemblance of the mature tertiary lymphoid structures that enrich with CD20 B cells, CD8 and CD4 T cells, and CD141 dendritic cells. In contrast to patients who have pathologic complete response, in general, non-responder display no tertiary lymphoid structures. Although immune cells such as B and T cells were observed, these immune cells were at the border of the biopsy or not infiltrating into the tumor core area. At week 6 post-neoadjuvant therapy, the non-responder showed B cell patches, but no concrete evidence of tertiary lymphoid structures. The non-responding tissue is filled with FOXP3 Tregs in green color, which may suggest an immunosuppressive environment. We next investigate if the number of tertiary lymphoid structures correlate with pathologic response outcomes in neoadjuvant immunotherapy. We found that overall patients with major pathologic response had higher number of tertiary lymphoid structures as compared to non-responders. However, we still observe heterogeneity in the data where patients with either good or poor pathologic response have absence of tertiary lymphoid structures. The next step is to further quantitate the tertiary lymphoid structures in their specific tissue regions, such as looking at the stroma or tumor core area, and to see if that can give us a better idea of the tumor microenvironment of the tertiary lymphoid structures. This study is still a work in progress, Although we have not done a comprehensive analysis yet, the results so far allow us to gain insight into the immune microenvironment in samples from clinical trials. We are able to observe hotspots of immune activity in responding patients. In contrast, in non-responding patients, we observe immune heterogeneity in different parts of the tissue, where in certain regions, it does contain little immune cells, but other tissue areas are immune deserts. We also observe tertiary lymphoid structures in patients who have pathologic response in general. We plan to further investigate these samples by performing cell-cell interaction, exploring immune cells in different parts of the tissue regions, and quantitative image analysis to gain further insights into immunotherapy. And last but not least, I'd like to end my talk here by thanking everyone who contributed to this research, especially patients and their families, all staff at MIA, resources at University of Sydney and Charles Perkins Centers, our collaborators and funders for their support of our work. And thank you very much for your attention.
Well, thank you, Camelia, for a great presentation. And with that, we can jump right into the Q&A. So the first question here, Camelia, how many additional time points improve insights into TME evolution during treatment?
I think that by including more time points in the study, we can capture how the tumor microenvironment changes over time, especially when the treatment is ongoing. The longitudinal approach of including more than 2 time points can help to track how the immune and tumor cells population evolve, helping to identify transient states or adaptations that correspond to therapeutic resistance or efficacy. I remember that a recent study published in Cancer Cells in 2024 by a group of researchers in Peking University in Beijing demonstrated how a coordinated cellular program with distinct response associated with immunotherapy outcomes. Their findings underscore that the compositions of immune microenvironment change dynamically, and the frequent time point sampling can improve understanding of the resistance and response mechanism that emerge over time. And this is also in line with our previous work published in Cancer Cells in 2019, where we revealed that the patterns in immune cell populations, such as memory T cells, that help to inform us if a treatment is working or if resistance might be developing in melanoma patients treated with immunotherapy. And by mapping this dynamic shift, I think that we could pinpoint windows of vulnerability where treatment interventions may be most effective, thus personalizing and optimizing therapy timing.
Yeah, that's a great point. Excellent. All right, next question here. What advancements in spatial multiomics could refine the characterization of immune excluded versus immune striving TMEs?
The advances in the spatial multiomics technology have changed how we analyze immune landscape within the tumor microenvironment in patients who are undergoing treatment. We can now see high resolutions of cellular heterogeneity and enable critical insight into immune excluded environment, which often refer to immune cells that are outside the tumor, versus immune-striving tumors, which is often marked by peri-tumor lymphoid aggregates, low infiltration of T cells, and the emergence of cancer-resistant subclones after therapy. These details help us to understand how some tumors keep immune cells away, which could help the design of new clinical trials that one can consider targeting the immune cells in the stroma area or enhancing intratumoral immune responses to improve the efficacy of immunotherapy.
Excellent, great answer. All right, here's a nice question here. What new biomarkers or therapeutic targets could be identified through deeper exploration of T cell memory state dynamics in resistant tumors?
I think that by examining the dynamics of T cell memory states in resistant tumors, especially through deep single cell molecular and spatial profiling, we could identify markers that tell us if the T cells are tired or still active. Preclinical studies have demonstrated that the exhaustions and reinvigoration cycles of T cells can influence the efficacy of immunotherapy in resistant tumors. For example, blocking the TIGIT pathway can augment the effect of pre-existing checkpoint inhibitors and reinvigorate tumor-specific exhausted T cells. As such, understanding these shifts in T cell memory states could inform personalized immunotherapy adjustment, improving cancer patient outcomes eventually.
Fantastic. Yeah, good answer. Okay, another one here. How do you see future studies explore the longitudinal effects of different immunotherapy combinations on TME evolution?
Well, the longitudinal studies can reveal how treatment shape tumor microenvironment composition over time. Studying how the tumor microenvironment changes can help us to understand and explore different combinations of immunotherapies. This approach could identify the optimum sequence of therapies and better predict how this therapy will interact and potentially improve patient outcomes. For example, studies like Prado neoadjuvant trial in this presentation offer a framework for exploring how therapy combinations shape immune infiltrations in the tumor microenvironment over a period of treatment and potentially allow us to understand the impact of durable response rates and helping us to identify when to modify therapy based on observed tumor microenvironment changes.
Fantastic. And how might further research on B cell signatures within lymphoid aggregates improve the prediction of immunotherapy responses?
Well, I think that further research on B cell signatures within lymphoid aggregates offer a promising avenue for predicting immunotherapy outcomes. B cells may play a crucial role in forming effective anti-tumor immune responses by presenting tumor antigens to T cells, and the distinct signatures could indicate the presence of supportive immune environments. I know that when I was reading a paper online by Helmick and other co-authors published in Nature 2020 showed that high lymphoid aggregates and B cell signatures were correlated with objective responses in cancer immunotherapy, especially in melanoma patients, and supporting the idea that robust B cell signatures within the tumor microenvironment can predict immunotherapy efficacy. And as such, recognizing these B cell expression patterns may help to identify patients who are more likely to respond to immunotherapy, refining patient selections and their treatment plans.
Great point. Very interesting. Okay, could expanding the patient cohort reveal additional melanoma subtypes or resistant pathways that weren't observed in the study?
Absolutely. I think that expanding the size of the patient cohort helped to capture the full spectrum of melanoma subtypes and resistant mechanisms that may be underrepresented in smaller studies. Further studies that include a larger group of patients can potentially identify rare, previously undetected resistant pathways and refine a cancer classification. This can guide the development of more personalized therapeutic approaches that target this unique profile from individual patients.
Excellent. Okay, what challenges were encountered with biospecimen variability, and how do you think future studies might address these?
That's a good question. Sample variability, including different protocols for sample handling and storage methods can introduce confounding factors that affect consistency and reliability of results. I can see that there are several published papers in Nature Protocols and Style Protocol documenting standard procedures for how samples are handled before and during phenocycler fusion experiments. In Australia, we have a user group meeting to discuss about the protocols to improve reproducibility in imaging data from patient biopsy or in vivo samples. As such, implementing standardized procedure and adding quality control checkpoints can help future studies to minimize artifacts, ensuring more reliable data for discovery and validation work.
Fantastic. All right, and I think in the interest of time, we'll just make this next question the last one. Camelia, how might T-cell receptor sequencing and neoantigen profiling refine our understanding of tumor-reactive versus bystander T-cells?
Right, that's an interesting question. I think that using T-cell receptor sequencing alongside with neoantigen profiling can distinguish tumor-reactive T-cells from bystanders by identifying specific T-cell receptors clonotypes that recognize tumor antigens. These techniques could reveal T-cell engagement within the tumor microenvironment, offering deeper insight into mechanisms of immune activations and exhaustion relevant to cancer progressions and immunotherapy response outcome. By profiling new antigens obtained from tumor, we can confidently identify specific T-cell receptors linked to tumor antigens that further clarify which immune cells are generally targeting the tumor and hence facilitating the development of novel drug combinations.
Excellent. Thanks so much, Camelia. Really appreciate your great insights today, both in your presentation as well as the Q&A session. It's really been a pleasure having you with us.
Thank you so much, Lem.
Yeah, our pleasure. Big thanks also to the audience for participating. And in closing, we hope you enjoyed this Akoya Biosciences and Scientist.com webinar, and we'll see you again next time. Have a great day, everyone.